Hello student, myself Dr.Shweta Humbarwadi, Assistant Professor from Department of Microbiology, CKThakur College, New Panvel. So today we are going to study about the capsule staining. So what is basically capsule? Capsule is the outermost part of some of the organisms which is protective in nature. Uh, yes, in many organisms or all organisms, cell wall is the outermost covering, but some organisms have one more coating which is known as a capsule. So this capsule is again acting as a virulence factor for the pathogenic bacteria. So we have to study about the capsule, or we have to check the organism will produce capsule or not. And this capsule staining was uh, performed by using Manvel's method. So what is capsule staining? So yes, and why there is a separate staining method used for the capsule observation. So unlike other cellular structures, these bacterial capsule do not radially absorb the most of the stains. So for making them appear as clear halos around the stained cell body, we are using here the negative staining approach. In negative staining, the cell will appear colorless and the background will be colorful. So the basic principle we are using here for the capsule staining is negative staining. So Manvel's method, which employs the Congo red, which is an acidic stain for the staining of background, and acid fuchsin as a counter stain which will stain the cell cytoplasm. So here the capsule will observe colorless, cell cytoplasm will be pink in color, and the background of the microscopic field is blue in color. So for capsulated organism, we can use this technique, and one of the example of these organisms are Staphylococcus, then Klebsiella, Streptococcus pneumoniae, etc. So what are the requirements for the capsule staining? So again, acidic stain, which is known as primary stain, that is Congo red stain, which is stain the background. Then Manvel stain, which contain the counter stain, that is acid fuchsin, which will stain the cytoplasm. Here we can use the capsulated organism culture like Klebsiella pneumoniae, Streptococcus pneumoniae, etc. Then S, clean glass slide. Then loop, needle, burner. Here again we are going to observe this staining over the light microscope under the oil immersion lens. Then for the cleaning purpose, or for the washing purpose, we will use the distilled water. So what is the procedure of this capsule staining? So first there are two parts of the capsule staining. First you have to prepare the bacterial smear with the Congo red, which is acidic stain, and then staining of this smear with the counter stain, which is nothing but the Manvel's stain. So in this, first you have to apply 1 drop of Congo red into the culture, bacterial culture, then prepare a thin smear over the slide, then again air dry it. After drying, you are going to add the Manvel stain. There is no water washing step over here. Directly you are going to add the Manvel stain. Again, heat fixation is not necessary. If you are going to heat fix the slide, then what happens? The capsule structure will get disturbed. So directly Manvel stain will be used over the smear. After that, 1 to 2 minutes, we can decant the excess stain. Again, air dry it and then observe under the oil immersion lens. Here is the result how we will observe the capsules of this organism after the staining. So here clearly it was observed that the cell cytoplasm is pink in color with a colorless capsule around it, and the background will be as blue in color. So what is the basic mechanism over it? As we know, we are using the Congo red in the staining, but still the background is bluish color because again it contains the acid in that. So Manvel's solution, which is the composition of different ingredients including stain as well as acid and the penetrator, phenol, etc. So because of this, what happens? The background, which is observed bluish in color because of the acidic stain, and basic stain will specifically stain the cell cytoplasm, and capsule will remain colorless. What are the conclusions of this capsule staining? Manvel's method for capsule staining is a valuable diagnostic technique with significant clinical applications. As we know, most of the bacteria which are pathogenic have the capsules, which include Streptococcus pneumoniae, Klebsiella pneumoniae, Bacillus anthracis, etc. And this capsule detection aids in understanding the bacterial virulence. So what is virulence? Virulence is the ability of any organism to cause the disease. So those organisms which possess the capsule, they are more pathogenic than the non-capsulated organisms. So after this capsule staining, we will get the idea about that organism's behavior. So this capsule staining is essential for diagnosing the infections caused by the encapsulated pathogens. Manvel's methods provides clear differentiation between the capsule and cell body. Understanding the capsule presence, which guides the treatment decisions and vaccine development. If the capsulated organism is there, then we are going to use different types of treatment methods. This technique remains a fundamental tool in a clinical microbiology laboratories for rapid bacterial identification and characterization. If the patient is suffering from different cup type of symptoms, then by observing the capsule over the organism, we can identify the type of organism present in that sample also. So yes, after this performing the capsule staining, we will get the idea about the nature of organism, and which is very helpful in the identification of organism also. So thank you very much.